Hello everyone. Today in this series, we are going to discuss the questions related to the topic photosynthesis. Okay. From last few years, the question is being asked from the physiology sections. So I have decided to cover few important topics related to the plant physiology also. Okay. Now let's start with the questions. First question is number of photon required for the evolution of 1 mole of oxygen in photosynthesis is what this is called: quantum requirement, quantum yield, rate drop, immersion effect, or none. You must know that nowadays, now in our atmosphere there is a photogenic this oxygenic photosynthesis, in which oxygen is released from during the process of photosynthesis due to the photolysis of water. You all know the general CO2 from the atmosphere and water from the atmosphere, and in the presence of sunlight, plant pigment converts this into starch or glucose plus O2. And this O2 release comes from the water in the presence of light. Okay, in photosynthesis, then how much photon is required to evolve or for the evolution of 1 mole of oxygen? That this is not the question how much photon is required. The question is that what is that term? What we tell this thing, if we have to tell that number of photon required for the evolution of 1 mole of oxygen in photosynthesis, that is what? Quantum requirement, quantum yield, rate drop, immersion effect. What is that? Okay, let me explain you. What is first? Let me explain rate drop. Rate drop is that this is the minimum when photosynthesis is minimum, means minimum photosynthesis yield when they are supplied with the monochromatic beam of 700 nanometer or less than 600 nanometer. Okay, this is rate drop. When there is enhancement in the photosynthetic yield when both 700 nanometer and 600 nanometer light is provided in mixed, both are mixed, and then they are both a range of wavelength of light are provided. Then there is increasement in the photosynthetic yield, that is known as immersion effect. Now this is the number of photons required for the evolution of 1 mole of oxygen in photosynthesis, that is quantum requirement. That is quantum requirement. And immersion calculated this value, this is 8. Okay. Quantum yield is number of oxygen evolved, in the when by 1 photon of light, number of oxygen evolved. Okay, it's a 0.125. Next question is, which of the following is feature of C4 plant? You must know about C4 plant. C4 plant, there in photosynthesis there is C3 cycle, C4 cycle. On that basis we call it C3 plant, C4 plant. C4 plant is also known as Hatch and select pathway, And from this section, in the recent examination FCI question was AGM question was asked that which of the following is Hatch and select, so Hatch and select pathway. Examples are sugar pecan, maize, sorghum, okay, salsola, amaranthus. These are the example of C4 plant. Okay, in FCI AGM option was, I think either it was sugarcane or sorghum. Okay, then in that C4 plant, what happens? There are some distinct characteristics. Means C4 plant shows crange anatomy. In this C4 plant shows few different characteristics like crange anatomy. They have a special leaf anatomy that is known as crange anatomy. Also, C4 plant tolerates high temperature. And they lack the process of photorespiration. They are photosynthetic more efficient. What is crange anatomy? Mesophils is not differentiated into pellucid and spongy tissue. Moreover, you say that cells of bundle cells arranged in a concentric rings around the vascular bundle. That is also crange anatomy. Cells of mesophil cells and bundle cells are interconnected by plasmodesmata. These are the characteristics of crange anatomy. After that, dimorphic chloroplast. See, yes, C4 plant possess dimorphic chloroplast means chloroplast of the mesophyll cells are small in size and granule. Granule means they contain grana, while the chloroplast of bundle cells are large in size and agranule. You must know there is presence of bundle cells as well as mesophyll cells, both in case of C4 cycle. Negligible photoresistance. Already I told you this point. Okay, then answer will be all. Which of the following is the characterized by the SO2 active stomata? C3 plant, C4 plant, CAM plant, A and B, all. First, you try to know what is SO2 active stomata. In this condition, stomata open during the night and remain closed during the daytime. Okay, and this is not in this characterized. It is not characterized in C3 plant or C4 plant. It is characterized in CAM plant. CAM plant means CAM plant. You must know in this. What happens in case of this is also crassularia and acid metabolism. This and in this case the like CAM plants, you can take the example agave, helio, euphorbia. These are the example of CAM plants. Okay, primary acceptor of CO2 is PEP. In case of CAM plant, OA is the first product. In this case, pepcase and rubisco both are present in mesophyll cells. Pepcase and rubisco both are present in mesophyll cells. Okay. Next, wasteful process of photosynthesis, which involves 3 cellular organelles. Okay. Then C2 cycle, C3 cycle, C4 cycle, CAM pathway, none. See, the answer is C2 cycle. It is C2 cycle is also known as photorespiration. Photorespiration. Why it is called as wasteful process? Because there is neither synthesis of sugar, nor ATP, nor NADPH. Rather, it results in the decrease of CO2. There is a release of CO2 while utilizing the ATP. But we have seen in case of normal photosynthesis, what happens? There is release of oxygen and production of ATP. Here, what in photorespiration it is opposite. There is a release of CO2 and ATP is consumed. So it is called as a wasteful process. About 25% of the carbon is lost during this process. And this process takes place in 3 cellular organelles: chloroplast, double membrane structure, peroxisome, single membrane structure, and a mitochondria, which is also double membrane structure. Okay, then this is known as number of carbon in the primary carbon dioxide fixation product in C4 cycle. Okay, see in C4 cycle there is an OH select pathway. The primary CO2 acceptor is a 3 carbon molecule, that is this phosphoenol pyruvate. First CO2 acceptor is in C4 plant is PEP, and the first stable product is OAA. And this is 4 carbon compound. Now, which of the following is photosynthetic inhibitors? Yes, of course, DCMU, PEN, diquat, paraquat, all are the photosynthetic inhibitor. Okay. In this, how they inhibit the photosynthesis? A few of them inhibit blocks the PS2 of the light direction, and this way they inhibit the photosynthesis. Okay, guys, thank you. See you in next lecture.